All right, hello everyone. Thank you for stopping by my talk this morning. So before I get into the nitty gritty of this, feel free to reach out to me on Twitter. My Twitter handle is listed here. And once this session is over, not just my talk, but the whole session during the next break, I'll be at the bench in the sunroom if you'd like to chat more about it. So I'm really excited to talk about my work today on genetics problem solving and how we can use trace data to better understand how students solve genetics problems. So first off, why genetics problems? Why are we interested in genetics? And a working knowledge of genetics is really important in today's society. So many of you might recall the Human Genome Project, and that's the first time the human genome was sequenced at a cost of about $3 billion US. And so it was hypothesized at the time that the cost of gene sequencing was going to follow Moore's law, which Moore's law talks about the changes in computing costs. And so although it did roughly follow Moore's law until about 2007, it steeply dropped off. So we went from about $100,000, which is already a pretty big decrease from the $3 billion of the Human Genome Project in 2002, to being below $10,000 in 2013. And it's now been below $1,000 for a few years now. So the cost of genome sequencing has dropped substantially. And as a result, it's easier than ever before for consumers to get their genetic information. So many of you may be familiar with 23andMe or Ancestry sequencing. You know, you just walk into a drugstore somewhere and you can buy one of these kits and find out your genetic information. I mean, what do you do with that? I'll come back to that in a minute. And it's not just sequencing that's coming on the market. There's a lot of new controversial genetic technologies that are coming about. So for example, 3-parent babies. So that is conceiving a child with a genetic material from 3 individuals. So this is currently legal in the United Kingdom. It's still illegal in the United States, although the first baby was born from this procedure in 2016. And also the science of de-extinction. This uses a neat new technology called CRISPR-Cas9 to edit elephant DNA to make it look more like mammoths and potentially bring mammoths back from extinction. And so as all of these new technologies come up, there's new room for consumer decisions, and there's a lot of ethical and policy decisions that now need to be made. And so a working knowledge of genetics is really, really important for society and for reaching overall genetics literacy. And problem solving is an important process to genetics literacy. So this is the process of finding a solution to an unstructured or complex situation. And genetics as a discipline relies very heavily on problem solving. And as I kind of mentioned in the introduction to this talk, this isn't limited to classrooms anymore. So you can get your genetic information and start making and start needing to solve problems in the real world. So for example, this is actually a figure from my book that just came out showing the Punnett square, which is a graphical representation of inheritance information of my husband and I and our potential blood types for our offspring. So problem solving across the board tends to be difficult for students, and genetics problem solving tends to be particularly challenging, and there's a few reasons for this. One is the necessity of using math, and there's a whole nother field that studies quantitative methods in biology and how that's difficult for students. It requires abductive problem solving, so being able to start at the end and work your way backwards, and also this ability to reason across multiple spatial scales. So that's being able to think about what's going on in the nucleus at the molecular level with my genes. How does that relate to what I actually see? And what does the environment have to do with all of this? So being able to integrate all these pieces of information and solve problems using it is very difficult for students to do. So what's going on in the brain during problem solving? So it's hypothesized that there's 3 levels of cognition that are in play during problem solving. So we have just regular cognition here at the bottom. So that is reading, gathering information, recalling information. And we have the metacognition. It's reflecting on what I know. It's planning on what I need to do next in this problem solving. It's monitoring where I am, and also the epistemic cognition piece. So metacognition is often thought about as thinking about thinking, whereas epistemic cognition is knowing about knowing. So how do I know I've reached a certain point in my problem solving? And metacognition in particular is theorized to be really important for problem solving, helping to link cognition to what I'm going to do next in this problem. The problem with metacognition, and epistemic cognition has this problem as well, is that it can't be directly measured. So thinking about thinking is not something that we can easily measure. So what do we do? We know it's an important process. So how are we going to do it? And so there's been a few papers recently about technical solutions, particularly in learning analytics, to measure these latent constructs like cognition. And so epistemic or metacognition, and looking at measuring these by student traces in some kind of authentic activity like problem solving. In terms of epistemic cognition, and in particular epistemological beliefs about science, that can be viewed through students' inquiry practices. So for example, this is a paper looking at inquiry trajectories of novices and experts. Only the novices are shown here as they work through an authentic science inquiry simulation. And these practices are actually found to be correlated with their epistemological beliefs as well as their experience with authentic science practices. And so the idea of using learning analytics for assessment of epistemological beliefs isn't new for the field. So it's been presented at the 2017 and 2017 LAC conferences using NLP of discourse during inquiry, and also K-means clustering of these inquiry profiles. So this is the work with epistemic cognition and epistemological beliefs. Now, how about metacognition and problem solving specifically? So self-regulated learning is a metacognitive process that can be measured using data traces. And so prior work has used trace data to examine self-regulated learning. So I just picked two examples from the field. In the first example, the authors used process mining techniques and sequential information to see that students who received metacognitive prompts during the activity had better transfer compared to controls. And in another paper, students were asked to generate models, and each group of students were given different levels of prompting. So one group of students had metacognitive prompting, another group of students had more content prompting, and the other group didn't have prompting at all. And then the different models that were developed were compared to experts using this HiNAT tool. Okay, so getting into the current study. So as I started with, the working knowledge of genetics and the ability to solve genetics problems is becoming increasingly crucial in today's society as all these new genetic technologies are coming up, and it's necessary for people to be able to reason and solve about genetics problems, not just in classrooms, but in the real world. Metacognition is a crucial skill for successful problem solving, but there's challenges with measuring it, but maybe we can use trace data to understand latent constructs like metacognition. So the big research question going into my work is, can trace data reveal cognitive and metacognitive processes during genetics problem solving? So this study involved 295 introductory genetics students. So these are undergraduate students in their first and second year of college. Majority of them were in some kind of biology major, although there was about 15 to 20% of students were non-science majors. So as part of the intervention, they were asked to complete extra credit genetics problems over the course of the semester. And while they were solving the problems, we asked them to think aloud on their processes while they were solving them. And then the outputs that we examined were, did they get the problem correct? How did they do in the genetics concept assessment of the GCA? So this is an established metric of students' genetics understanding, and also their final exam scores. So for our analysis, we used an emergent coding approach to identify 19 processes that we then binned under 5 categories. So what are these processes? So first we have concluding processes. So this is eliminating something. So we see a lot of these in pedigree type questions. So for example, this student is saying, you know, we can rule out that this is a sex-linked trait as a mode of inheritance. Or we can say this cannot be homozygous recessive based on the information at hand, or making a claim. So based on this cross, the chance of the offspring producing aldose would be 50%. The second category we looked at was metacognition, which included monitoring, checking, and planning. So for example, the student is planning by saying they need to reread the question first, and they need to figure out what the genotype of the offspring is going to be, or they're going to say, I'm going to compare the pedigree to the gel to see if there's any patterns. Checking is a little bit hard to see out of context, but this person looked at some kind of data and said, you know what, I still think I'm right. I still think I'm on the right track here. Or I want to go back and look at these pictures again and see what they're actually showing. Our next category is orienting. So if you think of somebody who's orienting themselves on a map, they are recalling information. So if we're using our map example, okay, where am I in relation to something else? Or what do I know about where I am? So for example, this student is recalling by saying, you know what, I remember from class that there's only a 25% chance of this happening. They could identify similarities between question types. So this question is similar to the last. So similar to if you're trying to navigate around, you might say, oh, this looks like where I was before. And also clarifying information. So this is a student saying, you know, and I'm going to read the question again, make sure I've got it. Our fourth category is reasoning, and this includes reasoning and justifying answers with evidence. So being able to say, I can conclude that the trait must come from the X chromosome and the mother must be a carrier. So they're citing some kind of evidence for their justification, or saying something like only producing aldose means there is a non-recombinant gene, so there's only a 90% chance that recombination will not occur. And then our final category is execution. So this is using information. So at the top, we know this person has this genotype, so he must have gotten it from his parents, making some kind of calculation. So the chances of this happening are 70% times 50%. Creating a visual representation. So you remember the picture of the Punnett square I showed a few slides ago. So the student is saying, I couldn't find a good place to start, so I drew out the Punnett squares, or restating the process. So they're restating the process of non-disjunction here. Okay, so based on this coding, we assign each process from the single letter code. So the C more refers to those categories I showed on the last slide: conclusion, metacognition, orienting, reasoning, and execution. And then we ended up generating strings, so sequences of these processes. And then we built counts of all possible action diagrams to capture these sequences. And then this was fed into a greedy stepwise attribution selection process using Weka to get the most informative features for predicting outcomes. And then for our clustering analysis, we used k-means clustering with Weka again. We built clusters using these bigrams features. And then we looked at how cluster assignment related to outcomes. Okay, so here's our clustering data. So shown here, these are our features. The count is referring to how many students fell into each of our clusters. And then these are our outputs here. So the probability that they got the problem at hand correct, the average score on the GCA, and the average score on the final exam. And then just as a note on timing, so again, they were doing these problems over the course of the semester. The processes we're showing here are based on the problems they completed, whereas the GCA and the final exam were given at the end of the semester. So we identified 3 distinct clusters using the elbow method. So our first cluster is also the smallest. You'll be able to see the rest of the counts in a minute. And these grayed out, these are the centroids here, and the ones noted in gray are the ones that are more highly represented in this cluster. So in this cluster, we see more of these metacognitive processes. So we see conclusions followed by metacognition, metacognition followed by orienting, or execution, or orienting with metacognition, reasoning and metacognition, execution and metacognition. When compared to our second cluster, this one seems to be more distinct in terms of the reasoning followed by conclusion statements or sequential execution statements. So we're not seeing quite as many of these metacognitive processes, but there's not really much of a difference. There's no statistically significant difference here in the probability of problem correctness. There's a little bit of a decrease in the GCA and final exam scores, but we didn't actually find that to be statistically significant. When we look at cluster 3, we're also not seeing a lot of metacognitive processes, but we are seeing reasoning followed by execution and execution followed by concluding. So these execution steps seem to be important in this cluster. And what's particularly interesting about cluster 3 is that these students had the lowest probability of actually getting the problem at hand correct. So this was a statistically significant difference. And then just as another note that I'm showing probabilities here, not p-values. So the probability was much lower. But overall, final exam and GCA scores were not different. So this is some kind of problem level phenomenon that we're seeing here. So to recap our first set of results, we identified 3 different clusters. Students in cluster 3 were less likely than students in the other 2 clusters to get the problem correct, and they had the least bigrams with metacognitive processes include, but they had a lot of execution processes. So this could mean that there's some kind of focus on doing at the expense of reflecting, and this is something I'll come back to in the discussion section at the end of my talk. So the next thing we wanted to do is ask, what do these bigrams have to do with these course outcomes? So again, those course outcomes are referring to, did they get the problem correct? How did they do at the end of the semester in the GCA? How do they do at the end of the semester on their final exam? So again, here is our features and our outcomes. And then the X's you'll see will indicate if these bigrams are a principal component at predicting each of these outcomes. So in terms of getting the problem correct, we see a lot of concluding bigrams. So bigrams that include some kind of concluding process is very important for getting the problem correct. We do see some metacognition ones popping up, so metacognition, conclusion, reasoning, orienting followed by metacognition or reasoning, reasoning and conclusion, and reasoning followed by orienting or reasoning as well. Okay, so so this is our proximal outcomes, so how about our two distal outcomes? So in the GCA, we see three processes that come up as important for both: sequential conclusion statements, metacognition paired with reasoning, and reasoning paired with conclusions. And we also start to see some differences as well. Again, these conclusion statements seem to be really important at the level of the problem, but not so much on the GCA. Whereas multiple metacognitive steps in a row seem to be really important on the GCA, but not so much at getting the level of the problem correct. So what about our other distal outcome? So again, sequential conclusion statements, metacognition followed by reasoning, and reasoning followed by conclusions were important for success at all three outcomes that we looked at. But multiple metacognition steps in a row and these execution followed by orienting or reasoning steps were found to be important for both of our distal outcomes. These are really important for, success at the very end of the course. But these tend to be more important for early in the semester and getting the individual problem themselves correct. So we have two kind of different outcomes that we're seeing. So two different sets of processes associated with different outcomes. So recapping these results, so different features are associated with different course outcomes. We see set A, which I've highlighted in purple. These are principal components for getting the problem correct only. The peach are for the summative assessments only, and the green are associated with success on both the proximal and distal outcomes. Okay, so one thing that's come up at LAC many times is how does learning analytics relate to pedagogy, and what does this mean in the classroom? So I've framed my discussion more around what does this mean in terms of pedagogical intervention. So different processes are associated with different success benchmarks. So making interim conclusions was important for getting problems correct, but it's not associated with success on either the GCA or the final exam. And then we note that these sequential metacognition steps or execution followed by orienting or reasoning were important for success on these summative assessments at the end, but not the individual problems. So could there possibly something with, is this a reflection of how well students understand what they're executing? So are they able to be metacognitive and able to think about, I'm going to draw a Punnett square and this is why it helps me solve the problem. And so is it really telling us how students understand how they're completing these problems? So possible classroom applications, we could do instructor modeling or importance on the emphasis or reason why we do these execution processes. So not just calculating because we think we're supposed to make a calculation because it's genetics course, but really thinking about what does that mean conceptually. And also kind of promoting this increased engagement with the material rather than finding the answer. So a lot of classroom students are just focused on, I want to get the answer, what is the correct answer? But some of these problems require a bit more thought about that material and a bit more reflection. And so giving students the opportunity to reflect and focusing on reflection might help them become overall better problem solvers rather than just saying, all right, well, here's my list of claims that I'm making. So it gets back at this shift from doing to reflecting, and incorporating reflection is an important part of equipping students with the skills to be able to solve genetics problems. So the other kind of interesting thing is these processes that we found that were important for all outcomes. And so these are the ones that were highlighted in green on that chart that I was showing you. So making sequential conclusion statements, pairing metacognition with reasoning, or pairing reasoning with making concluding statements. So there's something about the supporting process, the metacognition or the reasoning leading to a conclusion that is important for success in a genetics course. So it gets back to what I was saying on the last slide, is that you need to have this reflection piece along with your claims to really understand what's going on and to really be successful at problem solving. So possible classroom applications, again, so the role of instructor modeling. Or some kind of technological support that helps remind students to reflect and to orient and reason rather than just do the problems and get it done so you can turn it in. So it's kind of trying to think about how the instructor or other technologies could help support students to think a little bit deeper rather than just try to get it done as quickly as possible. So big conclusions, overall contributions of this paper. So it contributes to a growing literature on using traces to assess latent constructs like metacognition. It gives us some new insights into how students solve genetics problems and things that we can focus on in the classroom. And it identifies processes that are important for success on different outcomes. So it will be important for guiding pedagogical approaches from how do we just solve problems versus how do we help students succeed in genetics courses. And it's also a nice example of how we can use learning analytics to connect to classroom practice. So in closing, I'd like to thank my co-authors as well as some collaborators at the University of Colorado. I'm going to put a shameless plug-in for my new book. I mentioned this at the beginning of my talk. If you liked the bit on genetic technologies and ethics, please do look me up. I'm offering a discount during the LAC conference for LAC attendees. And with that, I'd like to thank you for your attention, and I'm happy to answer any questions.